Welcome to video 6 in our course G06 on transcriptome analysis. And now we're going to talk about why we have many different RNA Seq tools, and yet we get different results. So it's worth considering what approaches we take and what new information we get as we find out more about RNA Seq and different approaches, and there's always new information emerging. Well, one of the important messages from this course particularly is that there are better methods and worse methods to use. One of the common but less effective methods are the likes of EGR and DNA-seq, DSeq, DEC2. And the issue is that they have very high rates of false positive rates, and you can read more about that in that 2022 paper, where although the corrected p-value may be less than 0.05, for example, this actually corresponds to an observed p-value of 0.2. So that means that your rates of real false positives will be far higher than you might otherwise expect. So there are different ways of getting around this, but you should be aware that if you're looking at differential expression using these tools, that your false positive rates will be very high. In this study, what they did is they looked at 1,000 negative control data sets by permuting two labels with and without treatment for 109 different samples. And what they can see is that there are particular tools, DEC2 and EDGE-R, where you see very high rates of false positives. So you can see here, this is the number of permuted data sets out of 1,000 where the gene is wrongly identified as a differentially expressed, and this is the total number of differentially expressed genes that are found. You can see that DEC2 and EDGE-R find very high rates of false positive values, whereas a Lima model, Noisy, DearSeq, Wilcoxon, t-test all find far lower rates of false positives, and that's kind of really important to take on board. Another important finding from the study was that parametric style tests are actually okay. Note that Wilcoxon is non-parametric, and this is because it is better powered to distinguish ones with more changed expression from those who have less changed expression. So that's also worth noting. In our practical later, what we'll use is a tool called Sleuth, and that seems to have good power to detect true positives. What we'll do is import what we see in this graph is a plot of the false discovery rate versus the sensitivity. And when we increase our false discovery rate, we expect also to get more true positives, and sort of a balance in terms of a positive predictor value between these two. What you see is that the Sleuth has relatively higher sensitivity than the other tools. So you see Boom or Lima there is just below, and then you see DEC2 and EDGE-R are a bit lower. So that's also something to note. Other details that you can look at in this paper in more detail is to consider that changes in annotation will also affect what genes and isoforms are detected as differentially expressed. And you see an example here at the top where we have the log2 fold change versus the log2 p-value. I see that if we compare the annotations, you see that ensemble 92 and 98 are not quite symmetrical. They are somewhat symmetrical, but not entirely. And you see ensemble 92 and RefSeq, there's more differences, and the same for ensemble 98 and RefSeq. If we look at the aligner choice, HiSat2, the older historical tool, has different results to STAR. TopHat2, another older tool, has similar results to STAR and HiSat to an extent. If you look at then the quantifiers, the likes of HT-seq is still research valid, whereas future counts of cufflinks are older and less accurate. And you see that there are differences between all three pairs in these diagrams. So the annotation you're using, the aligner you're using, and the quantifier that you're using all matter. So what we're kind of getting towards here in terms of our thinking about this is that count-based quantifiers may not be effective for looking for differential expression in genes. And that's because we may have challenges in detecting overlapping genes where there's a sense and anti sense transcripted. You can read more about this in this paper. Even if we use stranded RNA sequencing, which is our typical approach, especially at the institute, that doesn't overcome this. So what we're trying to do is determine the isoform level by assigning different isoforms where the read was produced. And then we can look at the effect of the extent to which tools are accurate for doing this in other work. So this is looking at 240 GTEx samples. What we're looking at is the difference between the actual estimated counts per sample. So this is the difference, or the degree that things are wrong up here and right down below. So this is more accurate ones. You can see that Salmon, Callisto, and RSM are more accurate, and Synctia is a bit off, whereas feature counts in HT-seq is much less. So you see that as well. If you look at if we go from the gene level here to the transcript level, you see string tie is much less accurate too. At the same time, we find that in terms of the false positive rates, which is over here at this diagram, that feature counts and HT-seq have low rates, the same for Salmon, Callisto, and RSM. But they have relatively more false negatives due to overlapping features. So you can see there we have a similar pattern for false negatives. And then if we look at gene false negatives going from transcript false negatives, you see these are lower, whereas these are higher for the false negatives, and the gene false positives, you see HT-seq and feature counts are lower in that instance, but Salmon, Callisto, and RSM are consistently lower across all different factors that are of interest here. So you can see there's quite different results from different tools. If we consider that differentiated expression should not be affected if the ratios of counts are the same between different samples. However, that's not what we see. We see when we look at this across different genes, again, Salmon, Callisto, and RSM appear to be more consistent. And this is also present at the transcript level, where we see the same three are better performing. So that kind of tells us that the difference between the true and actual log2 fold changes are more accurate with these tools compared to the other tools. If we toggle read depth as well, we see that false negatives decrease with depth as expected. And this tends to improve most for Salmon compared to the others. So you can see the percentage of the original depth here. As we increase the depth, it goes down for Salmon, whereas for Callisto or SEM, it doesn't really make a difference. And if we look at the transcript false negatives, all kind of decline in parallel. And if we look at transcript counts, again, that the accuracy improves as we get more depth. Linked to this observation for different tools, we also can compare outcomes for bulk versus single cell RNA-seq. So you can read more about this in that paper here. So if you look at the log2 of the bulk profile, look at the distribution of counts for different genes compared to the single cell profile. What we see is that the correlation is mixed. And we see especially that there's a lot of genes with some expression in the bulk profile that actually are denoted as 0 for the single cell. The same is true in part for the single cell versus bulk, but less to less lower extent. So consequently, if we compare the delta between the log2 fold change of the single cell minus log2 of the bulk profile, we get this dip here, where we look at the average that for low expressed genes, we can expect to detect them well in the bulk profile, but we may expect that we underestimate or don't detect them at the single cell level. So consequently, we only really can compare bulk versus single cell for high and intermediately expressed genes and isoforms. And this is kind of confirmed when we look across 3 different studies here. They're all the same plot. We're looking at the bulk profile versus the average single cell profile. What you see is that the bulk profile tends to have a more accurate detection level shown by the red triangles here, whereas the single cell tends to underestimate those different values, and it might be quite mixed between different experiments, as you can see here. So that kind of suggests that we should be cautious about comparing single cell and bulk profiles. So that's the end of this video. Please look at the next video for more details on the practical.